Hi, welcome back. So by now, hopefully you've had a chance to look at the literature or look at what past scientists have figured out about increasing yields. Now, what you might find out is that there ends up being a limit or a threshold, and that might vary depending on what plant species you're looking at. So we're going to be looking at oats or a type of wheat. This is one of the major staple food products that are produced around the world. So that's why we're thinking of this might be a really good example or model to use. And you can find seeds or wheats pretty much anywhere where you have dry conditions but enough precipitation during a particular season. So in Australia, our wheat belts are usually found along temperate latitudes. So particularly in South Australia, there's a particular wheat belt where farmers find the appropriate weather conditions to grow their crops. Now, we're going to be using oats, like I said before. But before we kind of jump into just experimenting, we need to have a thought out, reasoned experimental design. Okay, so this is the part where you guys really get to nut out what it is that you'll be doing with your seeds. Now, the first thing that we'll need to consider are our variables. Now, you might remember there are different types of variables when we're thinking about an experiment. The first, what we'd like to call, is the independent variable. Now, this is the variable that we will be modifying as experimenters. So in this case, we're going to be modifying the number of seeds that we put into a particular pot. Now, this range of seeds, or from the very little to the very high densities, is up to us, but we need to consider what the carrying capacity or the threshold might be for a particular area. So for example, if we're using a small plastic cup, the size of say 250 mils, there might be a carrying capacity of 400 seeds. That might be the limit to which any seed will germinate. Now, obviously we don't want to jam pack our cups with too many seeds because we won't find any results. So we need to be realistic and kind of say, what's the most appropriate range? So when you're thinking about your independent variable, which is the number of seeds, you need to kind of come up with a range which will incorporate how many replicates we are able to do. And that comes down to resources and time and that sort of thing. Because at the same time, you don't want to be counting, because you've got to think, as a scientist, you're going to be making a lot of these measurements at the end. So you don't want to be having to measure 400 seeds per replicate. So it's kind of a cost-benefit situation here. All right, so the next type of variable that you want to be looking at is not just the independent, but also the dependent variables. Now, the dependent variables are those which will depend on what our independent variables are. So in this case, we'll be looking at the rate of germination, that is the number of seeds that actually sprout, as well as the biomass or yield of all seeds within a pot. We could also measure the number of seeds or the seedlings which grow to a certain height, as well as the number of seeds or seedlings that exhibit some sort of discoloration. These are all things that you can do, you don't have to, but you can decide on a number of different dependent variables that you're going to measure. Obviously, the more the better, but just consider that you have to kind of balance your time and energies wisely. And thirdly, the third type of variable that you'll need to consider is the standardized variables. Now, these are the variables we want to keep constant because we don't want some sort of rogue factor influencing our results. So we need to keep all other variables which aren't either independent or dependent variables as standardized. Now, it wouldn't take you long to consider that, well, what are some of the variables that we need to standardize? Well, maybe the conditions that the plants are growing under. So for example, we need to keep them under the same light give them the same amount of water, and we also need to make sure there's the same amount of soil or substrate within each of the replicates. Okay. We also want to make sure that even the substrate itself is the same. So in this case, we could either use a same amount of soil from a particular potting mix, or we could use cotton wool or something similar like that, just to standardize the variables there. Right. So now that we've gone through the different types of variables that you need to be looking at, we need to start considering, well, once we start growing these plants, how are we going to collect our data, or how are we going to measure our results? Okay, so if, for example, our dependent variable is biomass, well, we need to make sure that we're across how we're going to measure the weight of these plants. Okay, and well, in this case, you might want to consider getting an highly accurate electronic scales. We have a couple in our classroom, so that's not a problem. But the next thing you need to understand, well, how many plants? Are we going to measure the biomass of the whole plant, or just a part of the plant? So some people might say, okay, we need to divide the biomass as above ground biomass as to the biomass of the roots. Now that might be tricky. If you've got soil and things attached to the roots, you might need to be able to shake them off and dry them. Which makes another point. Are you going to be drying are you going to be weighing the dry biomass, or the wet or the moist biomass? Because these things can be different and have different results. All right, so if, for example, you wanted to measure the length of the plants, you obviously need a ruler or something like that. That's quite simple. But then again, you might need to consider how many individuals per replicate are we going to measure? Do we have to measure every single plant that germinated, or do we just measure a subsample? Again, it's up to you to decide. All right. Then finally, once you've decided on what are the variables that you're going to be changing, as well as the different types of equipment that you need, you'll then need to decide on what's called a sampling strategy or a sampling design. Now this just could simply mean, okay, after 10 weeks, we're going to harvest our plants and we're going to start making measurements. Or alternatively, you might want to set up some sort of weekly monitoring or sampling procedure. Now, you have to be aware that if you want to do something like this, that is a weekly monitoring or an ongoing assessment, not just a end sampling event, you need to make sure that you're not influencing the plants or the growth of the plants so they're remaining in the replicates. So you might then need to say, okay, I'm only going to subsample a couple of individuals per plot, or you might actually have different replicates which you set aside for completely harvesting at end of 2, 3, 4, or 5 weeks. Again, you need to consider time as a resource and how much effort you have time to put into measuring these different types of frequencies. All right, so that's basically all I have to say for today. However, we really want you to kind of nut these things out for yourself. So by the end of today's lesson, or by end of the week, I want to see that you've actually thought about all these things and come up with a potential experimental design or plan. Okay?